Okay, so now we're going to move to the next presenter, Professor Mathew Chang, who is a director of Singapore Consortium for Synthetic Biology and Technology Innovation. Mathew Chang is a director and a clinical and technical innovation, associate professor in biochemistry and synthetic biology, the Young Lee School of Medicine and Natural University of Singapore. His research focuses on studying the engineering of biology to develop autonomous programmable cells for grand challenge applications, supported by local and international organizations and industry. Particularly, he has a significant contribution to the development of microbial cells programmed to perform targeted biological functions with more than 100 applications. Let's welcome Dr. Chang. Thank you.
Thank you so much for having me here today, and many congratulations to Jeffrey and also all the organizers of this wonderful conference. So again, my name is Mathew Chang, and I'm from the National University of Singapore, and one of the organizations that I'm representing is Singapore Consortium for Synthetic Biology. So this is actually one of the 8 national technology consortia set up by Singapore government, in particular Singapore's National Research Foundation. And the main mission actually has been to bring the researchers together and also accelerate collaboration between academic members and industry members. So currently, all the universities of Singapore are part of the consortium, and we have over 20 companies as industry members of the consortium. So we've been actually making a lot of efforts to partner with other international organizations focusing on synthetic biology. We've been actually playing a leading role in setting up Asian Synthetic Biology Association, as well as a Global Biofoundries Alliance. I'm very happy to say that very recently our consortium and Synthetic Biology Africa, we actually had a joint event and a webinar together. In that event, Jeffrey actually gave a very insightful talk on synthetic biology in Africa, and also we actually really had a very, very good discussion how we can advance synthetic biology further together. And on behalf of synthetic biology community in Singapore, I'm really looking forward to deepening our collaboration and partnership. So as briefly introduced, my lab specializes in designing and also constructing and engineering genetically encoded circuits that can potentially enable living cells to show new cellular functions, ideally on demand. So we are studying how we can design and engineer genetic circuits of interest in a more systematic and predictable manner, and how the living cells actually respond to newly designed genetic circuits. So at the very upstream of our research, we design the entire microbial chromosomes and construct redesigned chromosomes from the scratch. At the bottom of this bottom-up genome engineering approach, in our view, can really enable us to better understand how genome works. But at the same time, at a more downstream, we also engineered microbial cells to function as microbial cell factories, which can produce chemical compounds of interest in a very sustainable manner. At the same time, we actually have been running in collaboration with clinicians in Singapore quite a few projects that aim to develop live biotherapeutics using the microbes that live in our body. So as we all know, our body actually contains a lot of microbes. So if you look at our body, every corner of our body, in my view, actually has a large number of microbes. So one question that actually my lab asked a couple of years ago was whether we could give new functions to the microbes living in our body so that they can serve as potential therapeutics. So this is our long-term vision, this schematic that I laid out on this slide. So we aspire to develop platform technologies where the microbes are living in our body and they be programmed or even engineered to manage health and disease in colon cancer, and also the production and as well as the secretion of the anti-cancer compound from food metabolites. So in this particular case, so as I mentioned to you before, so our strategy has been converting food metabolites originating from cruciferous vegetables, including broccoli and lettuce, in vitro as well. And our hypothesis, as mentioned to you before, was that engineered microbes could bind to colon cancer and convert glucosinolate coming from cruciferous vegetables into sulforaphane and leading to cancer killing activity, which we experimentally validated using in vitro cancer cell lines. So we got very excited by this in vitro outcomes, and then we wanted to move on to in vivo study using animals. So we established cancer animal models using a few carcinogen compounds. We also did a lot of genetic study to understand how long our microbes would stay in the gut. And then to make a long story short, we actually fed mice with about 1 billion engineered microbes once a week in a span of 14 weeks. And we also looked at the colonization in the bowel, and we also collected fecal samples on a weekly basis at both a 3-hour post-garbage and also 4-day post-garbage. And then as shown in this complicated graph, this data actually suggests that engineered probiotics and microbes that mice with supplemented diet actually had a reduced cancer formation, and at the same time, the vector bleeding, and the last graph, D, vector bleeding was partly recovered by our combinatorial approach. Just skip. So we actually also confirm the binding of our engineered microbes to colon cancer in vivo. So we actually resection colon tissues, and then we actually confirm the binding. So post-treatment colon tissue analysis as shown on this slide demonstrates that the mice with the engineered microbes and also supplemented cruciferous vegetable diet displayed a significant tumor regression and also reduce tumor occurrence, as you can see from this H&E staining data. And so collectively, in my view, this work actually demonstrates that engineered microbes can convert normal diet into a sustainable therapeutic regimen. In this case, we use this approach for treating colon cancer. And another study that I just want to touch upon is a more recent study that actually aims to eradicate Clostridium difficile infection. So in this case, we actually hypothesized that the dysregulation of microbiome-mediated bowel salt metabolism actually can contribute to C. diff infection. Therefore, its restoration can limit the pathogenesis of C. diff. So C. diff pathogens, some of you know, can germinate into more pathogenic form. But microbiome viruses were imbalanced actually taking place of antibiotic treatment. Therefore, in this particular study, actually engineered a genetic circuit in gut microbes so that they can sense antibiotic-induced microbiome biosis signal, and then limit the germination and also vegetable growth of seed pathogens. To make a long story short, that we designed and engineered genetic circuit, and we have to put this into microbes. And actually, as you can see here, in vitro setting, our engineered microbes reduce the toxicity of CD infection. And as you can see from in these images, we also move on to in vivo study, and this research actually shows that the microbes engineered to modulate intestinal metabolite can effectively inhibit C. diff infection in animal models. So hopefully, we're convinced that synthetic biology can also be used to engineer microbes, especially the microbes that live in our body, and these microbes can be transformed into potential live biotherapeutics. With that, I want to